This video we're going to focus on contraction, and this may end up being 2 videos, but I'm not quite sure. We'll see how it goes. So muscle fibers and neurons are considered to be electrically excitable cells. Their plasma membrane has a voltage change in response to stimulation, and this has always been something that's really hard for me to visualize, but I actually think the pictures in this PowerPoint and the animations are pretty helpful. So I would encourage you to watch over this more than once, watch those animations, and use the pictures. So let's be clear about what we mean. Voltage, which is also considered an electrical potential, is a difference in electrical charge from one point to another. Now, a lot of times we've talked about ions, but we know that that means that they have a positive or negative charge. But one ion doesn't feel like a big deal, but when we're talking about thousands of ions, then it can add up. So resting membrane potential is what we're going to go back to. It's about 90 millivolts, and that is maintained by the sodium-potassium pump. When we look at a resting cell, it is non-stimulated. There's extra sodium ions in the extracellular fluid, and then extra potassium ions in the intracellular fluid. They're both positively charged, but also in the intracellular fluid there's anions, which are negative ions, proteins, nucleic acids, and phosphates, and they cannot move through the cell membrane, so they are stuck in the intracellular fluid. There's more anions on the inside of the cell membrane than the outside, and so they make the inside of the cell negatively charged by comparison to the outer surface. So therefore, the cell membrane is electrically polarized or charged, which creates a resting membrane potential. So here's a picture that I really like. So this is the nerve cell or the neuron, and it's showing you inside the cell has those negative charges, and then outside the cell on both sides or on all sides is a positive charge. And so again, this is talking about the difference in those charges is the voltage. And so this voltmeter is showing you it's a difference of 70 millivolts. Now again, that's really hard to kind of quantify and kind of hard to picture, but just picture thousands of little ions all gathered in the cell and the inside of the cell that are negative, and then thousands that are positive outside the cell. So a stimulated cell, when the cell receives the stimulation, the ion gates open in the cell membrane. The sodium instantly diffuses down the concentration gradient into the cell. So it's going from outside the cell into the cell. Anytime we're saying down the concentration gradient, that means from a high concentration of that substance to a low concentration. He equates it to Black Friday at opening time, which is a great analogy. So all the sodium that is outside of the cell breaks on through and goes into the cell, and they are positively charged. So those positively charged ions overcome the negative charges that are in the intracellular fluid. And so the inside of the cell membrane briefly becomes positive, and that is called depolarization. Then immediately the sodium gates close and the potassium gates open. The potassium rushes out of the cell, and it's kind of repelled by the positive sodium charge, but mostly because of its concentration gradient, and so the loss of potassium ions turns the inside of the membrane negative again, which is considered repolarization. So resting membrane potential is a stable voltage seen in a waiting muscle or nerve cell, and then the action potential quickly fluctuates the voltage that we just described on the previous slide. And action potential is a quick up and down voltage shift from the negative resting membrane potential to a positive value and then back to the negative value again. So a lot of times if we're talking about a signal moving down a nerve cell, we're just going to call it an action potential. But really what that means is the negative resting membrane potential going to a positive value and then back down to the negative. When that action potential at one point on the cell membrane causes another to happen immediately in front of it, and so on and so on, it's kind of like a domino effect. So there's 4 sections to contraction and relaxation. So a lot of times we just focus on the contraction, but this book does a really good job of kind of giving you the lead-in, the two phases that get you there, and then what happens after the contraction. So a lot of other resources that you find might just focus on the contraction, but looking at all four is going to give you kind of a cohesive picture of what happens. So excitation is when the nerve action potential lead to the muscle action potential. And so we're going to revisit our neuromuscular junction again. Then the excitation contraction coupling is kind of the events that link that action potential in the muscle fiber to activate the myofilaments, getting them ready to contract. And then contraction themselves, where the muscle fiber shortens. And then relaxation. When all the work gets done, the muscle fiber relaxes and returns to its resting length. So let's look at each of those sections in more detail. So here's excitation, steps 1 and 2. Your pictures in your book are really good at kind of breaking this down for you. So the nerve signal or the action potential opens the gated calcium channels at the neuromuscular junction or at the synaptic knob of the nerve cell. So you can see those gates open and the calcium goes in, goes into the synaptic knob, and then it stimulates the exocytosis of the acetylcholine. So the calcium enters the synaptic knob of the nerve fiber, and then that stimulates the release of the acetylcholine from the synaptic vesicles. The acetylcholine is released in the synaptic cleft. You can see that in picture 2, and that's the little green dots that are released. And then 2 acetylcholines can bind to each receptor protein, and then that opens the gated sodium and potassium channels. So those are receptors on the proteins within the cell membrane of the muscle fiber, and then that opens the sodium-potassium channels. The sodium enters, changing the resting membrane from -90 to +75 millivolts, and then the potassium exits and the resting membrane potential returns to the -90. And remember that quick voltage shift, it's called an action potential, but when it happens right here, it's called an end plate potential. Okay, step 5 of excitation is when the voltage change in the end plate region opens nearby voltage gated channels, producing an action potential that spreads over the muscle surface. So the basically that action potential at the end plate then is kind of, again, a domino or chain reaction, and so is signaling the channels on the nearby, well, kind of down the cell membrane from that to also open. And so that is how a potential can spread or travel down a muscle fiber. So the step 6 and 7, now we're in the excitation contraction coupling phase, and that's the action potential spreads down into T tubules and opens those channels within the T tubules. When they open the sodium potassium channels in the T tubules, also the calcium channels are opening in the sarcoplasmic reticulum within the muscle fiber. So the calcium enters the cytosol. Again, this is all just kind of getting ready for the main event, which is the contraction. The calcium, when it gets there, it binds to the troponin in the thin filaments. And we said earlier that the troponin and the tropomyosin, they have a complex and changes shape to expose the active sites on the actin. So basically the arrival of the calcium here kind of makes the shape of the troponin and tropomyosin shift so that the active sites on the actin are available, are open and ready for business. I would definitely encourage you to watch any animations throughout here, and I'm going to pause here and pick this up in the next video.